humans, and there's a number of studies in humans that are worth mentioning to you. I'll mention two of them. One is a study of Ashkenazi Jews carried out by Nir Barzilai's lab. And he studied centenarians and found that centenarians were more likely to have mutations in the IGF-1 receptor gene that reduced its function. He showed in culture cells that the function of this mutant receptor is lower. And these people, this allele is more, or these alleles are more frequent in centenarians, suggesting that they play a role in the ability of these people to be young humans. And then FOXO, which is human DAF16, the variants of this are expressed, have been linked to exceptional longevity in populations all around the world. Look at this. In Japan, China, populations from the Middle East, Germany, Italy, New England, California. So this is by far the most validated so far of any of the longevity genes. So what this says, I think, is really important. It says, and maybe the reason this is so highly represented is because DAF16 can respond to so many different inputs. I just showed you that picture to affect longevity. So maybe the fact that it's networked in so deeply into this system, makes it, that much more important. But anyway, for whatever reason, it looks as though humans are going to be susceptible to the effects of this pathway. So in our lab, this caused us to start a new program using human cells in culture. So now we're looking for small molecules or RNAi clones that can activate FOXO3 and make cells more stress resistant in human cells in culture as a way of trying to take this further into the clinic. And I should mention, I'll mention this later, but these long-lived mutants are resistant to a wide variety of diseases, as you heard in the introduction. It's amazing. They're resistant in various organisms, to tumors, to heart disease, to various protein aggregation diseases like Alzheimer's disease, Huntington's disease. I guess there are others as well, but those are the ones I can think of right now. Diabetes. So it's really amazing. And a lot of these are age-related diseases. So it suggests that maybe if you went after aging itself, then you might come up with a pill that could be effective in keeping you young longer, but also in delaying lots of different age-related diseases all at once. Okay, well, we don't have that pill. So in the meantime, is there anything easy we can do? Now, I don't know the answer to this question, but I'm going to tell you that I think sugar just might be something to think about. Okay, so glucose, which is sugar, is known to stimulate insulin production in mammals. That's what causes the beta cells in the pancreas to make insulin. So insulin would rev up the activity of the DAF2 pathway. So could it be, that DAF16 is a little bit active in a normal worm, and if you completely eliminate DAF16, the transcription factor, worms live a little bit shorter. I didn't really point it out, they live a little bit shorter, they age faster. So is it possible you can make DAF16 more active by decreasing insulin signaling? Could you make it less active by increasing insulin signaling? Could that happen? So we wanted to ask that question. So we decided to just see what would happen if we gave our worms sugar. So the story is called the deadly sweet tooth. Let's see what's going to happen. And we gave them just a little bit of sugar, 2% sugar. A candy bar is 75% sugar. So this is not much. And sure enough, what we found was that glucose shortens lifespan by downregulating DAF16 FOXO. And it's been published, so I'm not going to go into all the details about it. But I want to just sort of touch on some interesting aspects of this. The first thing is we found this amazing result, which is that if we take a DAF2 mutant and we give it 2% glucose, we prevent it from living long. So here in blue, you see the long, beautiful long lifespan of the DAF2 mutant. And a wild type is, or not wild type, but so you can compare it to wild type in blue here. But anyway, the point is, if you add glucose to the DAF2 mutant, now the worm lives even shorter than normal. It lives actually about the same length of time as if it doesn't have a DAF2 mutant, it's just a wild type one, and you gave it glucose. That's one wheel, and you can see a similar effect with another wheel. So basically, if we had been growing our worms on 2% glucose, we would never have discovered the DAF2 mutant, because it doesn't really live very long. So this says a lot. It says food makes a big difference. And it makes you wonder about mice. So I didn't take you through all the various mouse mutants, but there's lots of them. And in general, they don't live as long as the worm mutants. The worm mutants live about twice as long as normal, or even longer. But the mouse mutants tend to live about 30 to 50% longer than normal. But mice are fed diets that have a higher glycemic glycemic index, that means that the sugar that's in the food gets into the bloodstream, fast, relatively fast. So is it possible that if you grew these mutant mice on a diet that had a lower glycemic, less sugar, lower glycemic index, would they live a lot longer? And also, what about humans? Humans become insulin resistant with age, and insulin resistance, which is at first accompanied by increased levels of insulin signaling, eventually leads to the loss of the beta cells into type 2 diabetes. It could be, and actually there are mutations in the insulin receptor that can cause type 2 diabetes. Is it possible that if a person with this tendency actually ate a really low sugar diet, that maybe they could have not only not get diabetes, but maybe have an even longer lifespan than a normal person? That's a really speculative, kind of crazy idea, but the only reason I say it is because that's exactly what we see in the worms. And we know, actually, that the effect of sugar on the worm is not that different. Well, it has a lot of similarities to the effect of sugar on humans. For example, I didn't tell you this, but the mechanism involves the expression of a glycerol channel. And glycerol seems to play a role in this decrease in lifespan. And the same glycerol channel that's regulated by DAF16 and sugar in the worm is regulated by insulin in humans. So I think that this is something to think about. So could a low glycemic index diet extend lifespan? And I personally immediately switched to this diet the day we found that the normal ones live shorter. And the DAF16, so DAF16 mutants, which already live short, they don't live any shorter if you give them glucose. That's one of the main reasons that we think that the reason that they're living short is because they downregulate DAF16. Okay, so let's just think about what is a low glycemic index diet. Now suppose you're dying from starvation. So you have a nice big meal and that's your last meal. This is what your blood sugar looks like. Your body does a great job. I've covered up what happens in the first 4 hours, just to surprise you in the next slide. But anyway, after 4 hours, and then until you die, your body keeps your sugar level about 100 milligrams per deciliter. First it makes sugar from one source, then another source, and when you finally die, you actually die because you start to degrade the proteins in your body. You die from that, not because you have not enough sugar. Your brain has to have glucose, so your body does a great job of keeping the glucose level really pretty constant. But look what happens after you eat a typical Western meal. It just skyrockets way up here. And if you eat a meal and then you wait 4 hours and you eat another meal, it just zigzags like that. So my goal is to try to keep my glucose level a little bit more down to this level, which seems to be a level that works pretry well for the body. It doesn't mean I'm eating no sugar. It means, I don't really know what it means, actually. We don't even know if this is going to make any difference in humans, but just so you know, I try to cut down on the sweets and starches in my diet. Okay, but now I want to tell you a story that may have an effect on you, if that alone didn't, which probably didn't. But anyway, okay, so here is a story about caloric restriction. So caloric restriction, or giving an animal less than it wants to eat, has been known to extend lifespan of yeast and worms and flies and rodents for a really long time. But for the last 25 or so years, there's been a big study going on in two places in the country, in the US, in Wisconsin and at the NIH, where rhesus monkeys are being calorically restricted. Now the results from the Wisconsin study are in and it's been published, and it seemed to work. In other words, this is the pathetic look of the monkey that was given all it wanted to eat. And it looks horrible, poor thing. And the food-limited monkey at the same time looks much better, it was disease resistant and lived longer. Okay, so that's what you would have expected. Okay. But now there's another study going on at the NIH, and they're getting really different results. What they find is that both sets of monkeys are living long. Those that can eat all they want, and those that are food limited. They live the same, and they both look like this. None of them have this disappearance. So what's different? Now this is the problem. It's going to take 25 years to check whatever your idea is as to what difference. But there's one big difference, which is that the monkeys in Wisconsin had a lot of sugar, sucrose in their diet, whereas the monkeys at NIH didn't. And the monkeys in Wisconsin got a lot of diabetes, which is connected to a high sugar diet. So this is an obvious reason. So it could be that in humans, caloric restriction, you don't have to calorically restrict yourself if you just cut down. Like if you eat your spaghetti sauce but skip the spaghetti, it could be just like being calorically